Thank you for joining the Once Changing the World, India's first Future Tech Meets Sustainability podcast. And today, I'm delighted and honored to have with me Dr. Anand Swaroop, a visionary in biochemistry and genetics, pioneering breakthrough in retinal diseases research. With a Ph.D. from the Indian Institute of Science and postdoctoral training at Yale, he has held prestigious positions at the University of Michigan and the NIH National Eye Institute. His Neurobiology-Neurodegeneration and Repair Lab leads cutting-edge studies in genetic regulation, photoreceptor dysfunction, and innovative therapies. He has contributed to over 350 publications. His work continues to illuminate pathways to restoring vision. Topic of discussion today is going to be how close are we to reversing blindness? So, Doctor, really appreciate you taking time and being part of the podcast. It will be great if you could start with your journey, from India to America, and why did you choose the field of blindness?
Thank you, Eddie. Thank you for having me. I'm just delighted to be with you and giving my first podcast. So I think the purpose from my mind is to let people know where things stand in case of blinding retinal diseases that are currently untreatable. I grew up in small towns in Uttar Pradesh, UP. I wasn't sure what I'm going to do. A friend of mine who was in Pant, he said, I heard, he was an engineer. He said, I heard that there's something called biochemistry. And if you do that, you can be in med school, a teacher or professor. I said, oh, that sounds very good. So I applied there and I got admission. And then I did my PhD with Professor T. Ram Sharma, who was sort of a pioneer biochemist in mitochondrial field. And he taught me the passion for science. And so I went to Yale. And that was, even though the salary they were offering me was 60% of what everybody else was giving, but I wanted to clone genes. That was the passion. When I went there, then I realized I'm colorblind. So it's a red-green color blindness, which is fairly common in males. 8% of males around the globe have that at different degrees. So I said, okay, the flies are not for me. So I want to do human genetics. And then I went to clone a gene for Duchenne muscular dystrophy in Professor Uta Franke's lab after working with Professor Garan. When I went there, somebody else cloned the gene before I could do anything. So near Duchenne gene, there was a gene for X-linked retinitis pigmentosa. And I was crazy for cloning genes. I wanted to clone a disease gene. This was in mid-80s. At that time, no disease gene had been cloned. Duchenne and muscular dystrophy and chronic glandular disease, these were the only two genes that were just identified. So I wanted to clone the gene for retinitis pigmentosa. I had no idea. Dr. Franke was moving to Stanford from Yale. I didn't want to go to Stanford. I stayed at Yale with Professor Weisman, and I told him I want to write a grant on this. He said, but you don't know anything about it. I said, yeah, I'll find out. So I just sort of wrote a grant with some new ideas, and it got 1 percentile. It was the top in the study section of that grant, and I became an eye geneticist. And since that, there was no looking back. And sort of it so happened that being colorblind and learning about photoreceptors and how we see color was one of the exciting part along the way. And then I realized that blindness is such an important problem all over. Most people are actually scared of going blind. Just after cancer, this is the most devastating sort of diagnosis that you can get. So I stayed in photoreceptor research and wanted to do something which is different and wanted to clone genes. And that's what I've been doing for a long time.
How much do we currently understand actually what blindness is all about? Why do we humans go blind? And what are the different type of a blindness?
Eyes are our window to the world, right? So in humans particularly, most of the things that we do in our life, most of the things we feel, most of the emotions, behavior, learning, a lot of these things are dictated by vision. So unlike, say, dogs and other animals which have very great sense of smell, some have very great sense of hearing, in human we use our vision. We use seeing is believing, right? We have this big saying. In fact, our brain, 30% of our brain is devoted to processing the visual information. Okay, and a lot more probably is devoted to responding to the vision. Okay, our memory, our learning, behavior, a lot of things are dictated by what we see, and of course all senses are important, smell and hearing and touch and everything. But at any given time when we are seeing, we are literally taking millions of points at any given time. When I'm looking at you, there's a background, your hair, your face, your shape, size, color. There's just so many different things. All of that information is going through our eyes into our brain for interpretation. But the vision begins with photoreceptors. Okay, but it's basically the light that information is actually sort of given through the light, and it's getting focused on the retina, which is at the back of the eye. But there's so many other components. So lens and cornea, they are optical element. They focus the light on the retina. If the light doesn't get focused properly, then you have blurry vision or you cannot see. So there are so many different parts. And then even if the retina is part of brain, is part of central nervous system, and it has got only few big major cell types, but there are literally hundreds of thousands of cells, actually millions of cells that are there, and they are working in concert. They're working sort of taking this information, integrating it, and sending it through an optic nerve to brain. Now, it's a very complicated process. You are capturing the visual information, you are integrating that visual information, you are doing some initial processing, you are packaging that information, and then you are sending it to brain. Now, it's the brain that interprets. Now, just imagine this billions of bits of information. We're talking about computers. Sometimes in the house, you have few wires, and if there's no color or there's no information given, we don't know how to fix it. In our retina, there are billions of connections. Brain is even more complicated. Okay, and now all that information at any different stage, if something gets blocked, something gets there's a problem, you can't see. Now, retina doesn't see. It only takes information sent to brain. So this whole system, the visual system, the part of brain that allows the interpretation of the visual information is also very critical. So literally this process involves billions of synaptic connections, the connections between different kinds of neurons. But even before the neurons, you have lens and cornea and so many other things that are required to put that information properly and send it to retina. So it's like, just think about it, that you have a very complicated computer that you have to build. But before you build the computer, you need all different parts. You have to get it from all over the world. You have to put it properly. You have to have some way of fixing it. Even that tiny little computer gets very complicated to fix if we are in trouble. Now, our brain is probably the biggest ever computer that has ever been built. It processes so much information. So there are so many things that can go wrong. So the blindness can be because of defect in focusing that information. You have lens, and if lens doesn't allow the focus it gets cloudy, you have what we call cataract. If the cornea cornea, which is there on top of the in our eye, on the lens, that's extremely sensitive. It has got millions of nerve endings. So you can remember, you can think that even a dust particle, if it goes in your retina, you have so much of pain and you want to get rid of it, you put water or whatever. Why? Because there's so much nerve ending. It's just so important. Then this is right in the front. Now inside, it's extremely complicated. There are cells like photoreceptors that will capture that light energy, and then they have the binary information, opening and closing of channels, which allows a signal to go to different other neurons, and those neurons then package, and there is just so much going on sort of in the background, and then they put bits together. This is for movement, this is for color, this is for this is for that, and all of the bits of information then is sent to ganglion cells, which again have their own factories of putting things together, everyone. And then the optic nerve then takes to brain, and brain now decodes that information, puts it in a proper image. So many steps. So now in the retina, you can have problem. If the photoreceptors die because of some problem, and there's a pigment epithelium that nourishes that. So if there are defects in there, you go blind. That's retinal degeneration. If there is a problem with ganglion cells, somehow they begin to die. You have glaucoma. You have, so there's just so many different kinds of blinding diseases that exist. So it's just so important for us. And so many different things can happen. So many different kinds of things that can affect our vision, and they lead to blindness. Something like cataract, now we can take care of them, right? We can make lenses, we can change them, and unfortunately in developing countries, in many places in the world, people don't have access to it. But thankfully in India, you have a lot of these wonderful pioneers, ophthalmologists, who many of them were trained in US, many of them they trained when they went back, places like Shankar Netralaya, and in Chennai, in Madurai, and in Hyderabad, L.B. Prasad, and many other places now in northern India. So all over India now, their students are opening these hospitals, and they're going to general population away in villages to do that. So cataract has become much more accessible. People can take care of it. Glaucoma also is a little bit easier to do, but not all kinds of glaucoma, but some you can take care, because you can try to reduce the pressure and all that. But retinal and macular degeneration, they are currently almost untreatable. There are some treatments for wet forms of macular degeneration, but other forms are really very hard. There is one gene therapy treatment that has come up, but it's sort of very not only expensive, but it doesn't work every place. So the biggest incurable blindness right now is because of retinal and macular degeneration. Right. But they are not the most common one. The most common is cataract and glaucoma, because people just don't get access.
Yeah. So, Doctor, thank you for kind of describing our vision, the reasons why we get blind. In today's world, I think, almost the entire new generation is so hooked onto the digital device that I see young kids as young as 3 years, 4 years, having to wear glasses. Unlike our generation, we didn't have these TVs and internet and so on and so forth. But the kids nowadays. So yes, and you pointed out that our vision takes somewhere around 30% of all cognition, which comes as a surprise. But yes, I think of all of five senses: sight, sound, touch, smell, taste. Sight is that sense which we cannot do without. And the way you were kind of describing the visual system, for me, I thought you were speaking of almost like a computer vision system, because the computer vision systems, today does pretty much the same thing. So is there some similarity over there between a computer vision system and or or the human vision system? And if you can share about your lab, the Neurobiology-Neurodegeneration Repair Lab, and your work in the fields of blindness and your research program.
Okay. So this is again a very loaded question, but let me just sort of, the computer vision that you are talking about, I think still has not reached even a fraction of what happens in our own tiny little eyes and in brain. It's just extremely complicated, but we are getting there. So computer vision maybe someday will reach there. But I think just imagine that we have billions of wires. They are connected in an appropriate manner. Every cell that is there in our retina, I mean, from photoreceptor to the ganglion cells, and I'm not talking about brain, I'm talking about just in the retina that sort of initially captures that information. So when light gets focused onto the retina, it goes through the whole width of the retina and goes to photoreceptors. Now, photoreceptors, as the name will indicate, they receive the light, and they have millions of molecules of a protein, a visual pigment. Now, there are two kinds of photoreceptors. One are called rods, and the other are called cones. And rods allow us to see in the nighttime. Now, cone photoreceptors are only small in number, and there are three different kinds in human. Depending on the visual pigment they have, there are short wavelength, medium wavelength, or long wavelength, or S, M, and L in short. These three kinds of cones allow you to see during the daytime. They are responsible for color. They are also in the center of our retina, there's an area called fovea. That's where the light gets focused. In the center, where there are more cones, there's the highest visual acuity. So these two photoreceptor rods, one would wonder, why do we have so many rods? Right? We are not nocturnal animals, we are diurnals. And so my lab has been working on photoreceptors primarily. And our goal has been to understand how these photoreceptors, rods and cones, are formed during, our birth, right? How from stem cells, how from it, just imagine how from a single zygote, from mom's egg and sperm, the whole human being comes up. We are focusing on that just on one tiny part. We are focusing on from that zygote, how brain, not even brain, retina, but not even retina, how photoreceptors are formed from those early primitive cells. And then we try to see how these photoreceptors change, how they respond to different conditions, light and other conditions, to age, how they change with different diets and whatever is going on. And then what happens if there is something wrong, if there's a disease, how they respond, how they die, what happens in aging, what is the impact on aging and disease. If photoreceptors die, you go blind. Retinal degeneration is the term we use when we have damage primarily or death of photoreceptors primarily in the peripheral retina. Okay, so we can see we have a big field of vision. Center focus, I'm focusing on you. Okay, so right now on this, so that's where I'm using my center fovea. But I have peripheral retina. I have a macula slightly bigger, peripheral retina. I can see on both sides. I can see my fingers even, my broad vision, right? So that's a peripheral retina. So when I lose photoreceptors in the peripheral retina, I get constricted vision. I have central vision only. Just imagine if I can only see in the center. So all this vision is lost. That is called retinal degeneration or retinitis pigmentosa, or retinal degeneration can be many different kinds. Macular degeneration, when we say, that's the degeneration of photoreceptors in the central part of the retina. So if that area gets cloudy, now you can imagine if you can't see in the center, but you can see in the periphery, then I can't, if that area gets blurry, then I can't see your face. That means I can't read, I can't drive, I can't watch TV. So right now there's one therapy that is out there, Luxturna, for a mutation for disease LCA, Leber-Condé amaurosis, which is caused by mutations in RP65 gene. Now that gene, you replace that gene, and many of these kids can see now, even though they were blind right from birth. But that doesn't happen in everybody. And you have to treat it at certain time only, so maybe, and that treatment costs, I don't know, $700,000, $800,000 and all. I don't know how many people can, I think, afford that. And gene therapy right now is being tried for many others. And RP65 was sort of relatively easier because it affects pigment epithelium, but things that affect photoreceptors and all are going to be a lot more difficult because those are neurons. But what our lab is doing is trying to find small molecules, drugs that can alter many of these pathways and maybe restore some sort of network, that disruption, and that way, so that way the cells can survive for longer time. We are not trying to bring the new cells like in stem cell therapy once cells die. What we are trying to do is, before cells die, if you can stop it. So we are trying to identify small molecule drugs which we can put in, say, eye drops, so that they can go in the retina, and if they are having bad effect, you stop. If they are having good effect, so we can test them and we can adjust the doses, we can do. So that's our goal, because those molecules then can treat many of these retinal degenerations, right?
Right.
That's what we would like to do. So we have been doing some screens, and we have some molecules. And not only we, but there are many other groups around the globe have begin to realize that a small molecule therapy that can sort of in a more generic way target many of these genetic defects will be great. So that's for retinal degeneration. Genes are like bhagya, luck, right? Bhagya is whatever God gave us, if you want to believe in God, or whoever parents gave us. They're the genes. We have little control over it. In general, genes is bhagya. Age is kal. Okay? Everybody is going to age. You have no control over time or kal. Environment is karma. So bhagya, kal, karma. In our Indian culture, we have these three that are very important entities that govern us.
Right.
And they're all, everything that we do are based on partly on that. What is karma in this case? Environment? Karma is what we do, how we eat, whether we exercise or not, whether we sort of protect ourselves from UV or something, or we jump around, or whether we smoke, or whether we, so everything, that's your karma. The only thing that you can really do is control, to some extent, your karma. Sometimes you can't do that because genes dictate your karma as well, and age dictates your karma as well. So they're all related. But karma, at least you have some control over that. Genes, age, and environment is the interaction of these that really governs how we get macular degeneration, or for that matter, almost all complex
Exactly. Right.
phenotypes.
Right. Right.
Okay, whether it's
Right.
diabetes or everything else is based on that. So
Right.
there, our focus is less on gene therapy, or my own focus primarily there is trying to reverse aging and environmental impact to reduce the impact of genes. Age and environment work by changing this epigenome, and epigenome can influence genome.
Right.
So if we can take care of our karma or environment, we can reduce the severity of phenotypes. We can reduce the impact of bad genes. We can slow down the progression. We can delay the onset of disease. Disease may come, but now if you have age-related macular degeneration, you're going to go, suppose, have the disease at 70, 75 years of age. If I can delay that to 95, I may die of other things before that, I get the disease, right? So won't it be wonderful if I can delay it by 20 years?
Right. Right.
So the
Yeah.
easiest thing there is diet, because you can control your diet. Easiest thing there is not to smoke. That's what I'm trying to do as a person who is susceptible to macular degeneration. But there could also be drugs, and we and others are working towards that to see if there are some, again, drugs that can slow down that impact of genes was given.
Right. 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 So, doctor, I mean, you said a whole lot of things, and I want to kind of unwrap that for my audience. You mentioned that your main work is on photoreceptors, rods and cones, and macular, as well as retinal degeneration. And you pointed out that there's three most important pillar of why we age. It's the genes, it's the age, as well as the epigenomes, which is the environment. What you talk about the epigenome, the karma, it's the epigenome, because it's not just the genes which dictates us, but it's also the epigenome, the environment, like if we smoke, and things like that. You mentioned your approach is largely towards molecular therapy, not so much of gene therapy, because gene therapy in itself is complicated process, and it's a very, very expensive process today. I mean, gene therapies are like, it goes in $1 million. There are others like Elon Musk, who's working with Neuralink, and then Max Hodak, who's working with a complete different approach to also tackle blindness by birth. How do we, I mean, you mentioned, obviously, I mean, if we kind of take care, do exercise, eat well, reverse aging, less smoking, these could help in reversing your age, and then maybe helping your photoreceptors not die. But what are the cutting edge of treatment in the space of reversing blindness, I mean, which is by birth? I mean, do we have something that can give us hope?
Yeah. So I think that's a great question. It's not only by birth, but also there are a lot of people who are going to go blind even if at age of 40, 50, 60, 70. Now, many of these people may or may, in the absence of any simple treatment, they may not want to try gene therapy, right, or something else, because there's some sort of risk they may assume and say no. So people are still going to go blind because of various reasons. So how can we help them? There are many ways people are trying. One way is to replace those cells that are dying by using stem cell derived either photoreceptors or something like that, which is going to be very complicated because forming those connections is going to be difficult. Second thing that people are trying to do is what we call optogenetics. So what you can do is you can put in the cells that are not dead. So if photoreceptors are dead, but your inner retina neurons, they are not dead yet. So could be bipolar cells or other cell type. Now you can put these visual pigments, some, from different organisms. They are non-human kind of pigments you can do. Those pigments can be activated by light or by some sort of signal, right? And then they can initiate the cascade of events in those cells, and those signals can then be amplified and can go to brain. Now, just remember, retina does not see. It's the brain that is interpreting the signals. So you can put in ganglion cells, you can activate those optogenetically, the optogenetic molecules, and then you can see something. But if it's in ganglion cells, the resolution will be very low. There's a huge amount of amplification of signals from photoreceptors to ganglion cells. I told you this complex network that amplifies every signal and gives you very fine resolution. That will go away. But you can see some light. If you put it in cells before ganglion cells, say in bipolar and all, you may be able to have some amplification and some resolution. In many of these cases, what people are trying to do is trying to see what kind of light may be needed to activate these molecules, right? Some of that may require red light or infrared light, some may light a little bit blue or whatever. You don't want UV light and blue light too much because that can damage other. So there's a lot of science involved in trying to figure out what kind of optogenetic molecules might be needed. And there's a lot of research going on. And in Europe, there was a trial in which one person particularly did get, they had two patients, and one of them showed some response to light and was able to go through the maze and see something. So many of these techniques can give you some vision, not resolution. You can see some light, or you can see where there's the obstacle. Now, computer vision is very interesting. Let's come back to what you're saying about how people are trying to see, or neural vision or whatever. I told you it's the brain that sees, right? So everything, in fact, the signals that go from photoreceptors to the next level of neurons, secondary neurons, to the tertiary, to later, to everything, there is a coding. They're all the electrical signals, but they're mathematically, you can interpret those signals. You can see how much amplification could be there. You can look for code. There are groups of people, including, say, New York and many other places, who have been trying to decipher the computer code of how we are seeing, right? And then you can make brain, you can give the code to brain so that you can now sort of figure out what is going on. Now, people are also having different kind of prosthesis devices. You can have glasses which sort of can allow you to have some vision of some kind, right? So there are so all of these things that are going on, they at this stage they are in very early primitive. There are things called Argus 2, and there are different groups of engineers that are involved in it. One of my friend Mark Humayun at USC has been big. His parents came from, his father, I think, is from India, Mark Humayun. So he's very well known, very top notch. And so he was very much involved. He's an engineer as well, an MD, Ph.D. and all that. So there are a lot of people involved in that. There are a lot of electronic engineers, a lot of different kind of, they are looking at from different angle. At this stage, it's a little bit primitive. What you're trying to do is you're trying to give signals directly to brain, but those signals may not allow you to distinguish among different features. When I'm looking at you, I'm looking at every hair that I can see, and and different colors of your hair, different facial features and all that. At this stage, and I can see the background, at this stage, they those kind of devices allow you to basically have some idea that the light is there and the light is moving around, but it the resolution is not there. Now, yes, maybe in 10, 20, 30. So I'm not an engineer, so it's very hard for me to sort of explain and see how. I think biology will go faster. I mean, I'm a biologist, sort of.
Right.
I think I think everything is mathematical, but in my opinion, that mathematical thing that is going on is so complex that I don't think even Watson, this supercomputer and all that, are really as complicated as our brain is and as complicated even our retina is. We don't understand any connectivity of billions of these little connections that are existing in very tiny, tiny area. Can we do that one day? Yes. I will never say never, because in my own lifetime, when we had no human disease gene cloned and we didn't have any idea about genome, we never thought we'll be able to sequence genome, thanks to computers as well. So the beauty is this huge interaction right now going on between biology and technology, or biotechnology, if you want to call it. And that the technology is moving very fast.
True.
Okay, much, much faster than biology is moving. Okay. The trouble is that unless you know biology, the technology can be over there and then they're not talking to each other as well. Okay. That's my little bit worry, because you have to validate these things. And so many times, why do drugs take long time to come to market? You do it in mice. Mice are not human. Okay. They tell you certain things, but they are very, so, and each gene and each mutation you have to test, it takes forever. So even today, after 30 years, we still are using some of those primitive techniques. Now, organoids have made it a little bit easier. You mentioned in one of your, and there are things that are going on that make it faster, but we still have the pace of testing the biological experiments has increased, but it's still way, way slower than the way the computers and technology are moving. And that's we don't understand the biology inside. You can do all kinds of things, but you need to connect it to what is inside here in order to have that high resolution. So I think that technologists need to devise ways to test biology in a faster way. If you want so you can create all computers, you can do all that, which our brain can adjust to and all that, that's fine. But understanding every biological reaction, we need to find some way of testing it. Right. Now, you can test it computationally, but still it's not real, right? In the real life, you need to figure out that it works. I'm not going to take a drug or something that has been just sort of computationally, yes, it must work, but I don't know whether it'll work in real human. I don't know if I want to do that, right? So as a human being, I'm sort of scared of doing that unless that becomes, so at least initially, is it moving faster? Yes, there has been, I can see it is happening, but pace is there's the logarithmic differences basically.
True. Small, tiny. Yes, exactly, doctor. So I think, the pace of technology is accelerated so much, and that's the reason we have these truly technologies that can really add value. But you rightfully pointed out, unless and until we do not understand ourselves, that means our cognition, as well as our genome, which you mentioned, 3.4 billion base pairs of DNA, and which brings out and makes us us. I think unless until we don't understand 80 billion neurons and 100 trillion synapses, the way they fire and wire, how they give rise to a sight, sound, touch, smell, taste, I don't think we'll be able to kind of make like really true progress. But yes, maybe somewhere down the line, if there is a convergence of sorts where maybe AI and scientists kind of find ways to kind of you know help each other and accelerate our understanding of who we are as a human, and then I think maybe we'll be able to make really progress. You alluded to retinal organoid, that you've been working on retinal organoid for disease modeling and developing of therapies. Could you maybe, as my last question, talk about retinal organoids, and also maybe your approach, the molecular therapy or gene therapy. When do you think this will be accessible for everyone? Because I mean, like you might mentioned, I mean, sight is the biggest problem, and do you think that maybe in what timelines do you give that, maybe you will be able to kind of reverse blindness?
So retinal organoids are basically retina in a dish, right? So we have been doing a lot of experiments earlier in model organisms, whether it's zebrafish or mice or something else. The main problem has been they are genetically homogeneous, and I told you humans are not. We had a tremendous variability, and so it's very hard to model that. So to model that a human disease in a dish. Professor Sasai, he passed away some time ago. He was the one who first developed these from stem cells. He by changing media and conditions was able to create a retinal structure in a dish. And since then, and that was about 12, 13 years ago, and since then, a lot of progress has been made, and people can see some function, electrical signals, and all that. It is still more morphologically sort of, it can recapitulate some of the developmental events, and it can recapitulate some of the disease conditions, because you can make stem cells from patients, and you can do from normal control individuals, and then you can try to look at how the organoids differ among that, and then you can develop assays to find treatments. So this is so I think that sort of recapitulates to some extent the human system, and we think to find any kind of treatment, we need to use both in vivo systems like in mice or something, and we need to use something in dish like organoids, and of course some maybe computational tools as well. So I think so what we did, we have been doing is using these organoids and photoreceptors that are present in these organoids to see, number one, how they develop and how they become defective in patients. So we took some patient organoids where the photoreceptors were dying after some time, and then we said, okay, how can we save them? And so we did large high throughput screens of known drugs, and we said, all right, here you are. We after a lot of screenings and further experiments, we found few molecules that seem to protect these photoreceptors from dying. So then we did some secondary assays using mouse models, and we realized that even when we inject these drugs in the eye of these mice, we were able to protect the photoreceptors. The mice who had the mutation and who were going to have blindness, well, we were able to sustain their visual function. So that was very exciting. And now then we have begun to test how these molecules function and what their role might be. And then we are beginning to use these molecules now to look at different types of what do you call the different animal models. So I told you there are 250 different diseases. We tested initially for one disease. Then we said, okay, can we do it for second or third? And now we have found some molecules that work with multiple models. And that's very exciting. And we are doing that. There are 3 or 4 other groups in the world that I know they have found different type of molecules that seem to function. So our goal now is to see if we can develop these molecules into eye drops and deliver them through that, and can it reach retina? So we are now beginning to do some assays to see how much drug is going into the retina and how much is needed to protect those photoreceptors. So there are a lot of these kind of things that are in progress. But one of the problems that we face many times is that big pharma is not very interested in repurposing of drugs. So what repurposing means is that you already have some drugs that have been approved. Now you use it for different purpose. Big pharma likes to have billion-dollar drugs. They don't really care about few tens of million. Now, even if now these drugs are repurposing for repurposing, right? So they can be tried much more easily for human disease. But to do that requires certain amount of fund, not too much, but so being at NIH, we are doing it, but it's slower. So we have been trying to look for partners we can have some smaller amount of funding who can really push it to do pharmacokinetics. And I'm not a sort of pharma person, right? But we will do it because we want to treat patients. We want to really go to see, can we, whatever we see in mouse and in organoid, can we replicate that in human? And because it's a repurposing drug, so it's supposedly safe. And eye is so far out that it's not going to go inside very well. So we have good protection. So chances are fairly high that we can test it very easily. So if we had some partner from this thing, it's much easier. But it's very hard to convince bigger companies to do that because they're not going to be too much money in this, right? So, but for us, that is, we don't care about that. I mean, I don't care. I mean, I have a bunch of patents, but I don't care about those patents. If I can help even few kids see, that's what my life's mission is done, then I can die peacefully. So when you say how long it's going to take, so we're trying to convince some people. There are smaller companies and other people who might be interested. So through your podcast, maybe some venture capitalist, somebody wants to put this thing and whatever. But I think we want to partner with somebody who will go much faster. If all goes well, if I can get a partner, we could have it out in clinical trial within a couple of years. But I think that's where the issue is for repurposing drugs.
Right.
So we'll see. We are working towards it. We'll see. But clinical trials could be as quick as 2 years, I'm hoping. This is a letter that came to me. I mean, that was given by a little girl, 6-year-old girl. Her name is Amy. That was some 10 years ago or so in one of the meetings. And she was 6 years old, going blind because of mutation. And this is what she said. Thank you for fixing my eyes. If you don't, I will still be happy. But please try. Love, Amy. Yeah. So what I want to say is that this 6-year-old girl was so wise that even if you don't do it, I will still be happy. How many people, adults, can say, but please try? And that's what this letter is in my office, and we see it every day. I talk to everyone. This is what motivates you. This is what it is all about. This is what we do, why we do it. And thank you very much for having me, and I appreciate it. And hopefully we'll meet again someday. And
Thank you, Doctor.
send me this, and I'm hoping it'll reach few people, and maybe someone will help us out in some way. Maybe.
Right. Doctor, really appreciate you taking time and being part of the podcast. More kudos to you and your entire team at your lab to keep on pushing. And you pointed out there's so many challenges. It's the pharma industry, which is, I mean, obviously profit at any cost. That's the way they function. I hope that, VCs who are listening maybe, joins in this, because vision is the most basic thing. And if with any senses that we lack, maybe we can still sustain. But without vision, I think we get completely lost. So I hope your work for repurposing drugs to your eye drops to the gene therapy, I hope that this becomes more accessible and for the larger population in the world. And because I think if we manage to reverse blindness, I think it'll be one of the biggest problem that we'll be sorting out. And yes, there are lots of people. I mean, besides you, like I mentioned, Neuralink is taking an approach of neuroprosthetic, where they're developing a brain-computer interface that bypasses damaged eyes and optic nerves and directly stimulates the visual cortex. So yes, maybe one of these, whether it's gene therapy, stem cell therapy, eye drops, repurposing drugs. I hope some of these kind of comes together, binds and sorts the problem out for the people who are actually in need of this thing. So really appreciate you taking time and being a part of the podcast. And my listeners, if you like what you see in here, then please press the subscribe button. And until next time, see you guys. Thank you. Thank you, Doctor. Really appreciate this.